All right, everyone. We are going to get started. Thank you all for being here at our Science on Tap. This is our last Science on Tap of 2022. Woohoo! Yeah. Just a reminder, this is a hybrid event, so we have almost as many people, in fact, maybe a little bit more online as well. So welcome to those folks that have joined us that way. If the folks that are online have any questions at all, you can go ahead and put them into the chat box, and Roseanne is our volunteer. Yay, Roseanne. That will help answer any questions that you might have if you're having technical difficulties. Also, for folks online, if you have questions for our speaker, go ahead and put them in the chat as well. Your cameras, mics, and screen shares are disabled in this web format, so your chat is the way that you can ask your questions, but we will read those questions out at the end and we'll have our speaker answer them. For folks in the room, yay, welcome on the stormy night in Newport. My name is Cinnamon Moffett, and I'm the research program manager here at Oregon State University's Hatfield Marine Science Center, and I am your host. Wanted to just let you all know that because this is a hybrid event, we're going to do the same thing where you all will need a mic to ask your questions. So we're going to do questions at the end. You either can go to the mic stand that's off to the side or just raise your hand and I get to run around and get my exercise and I'll bring you a mic. So thank you for that. Wanted to do just a couple of quick announcements. The next public event we're going to have is in January. So we typically take December off for the holidays and travel, but in January, we're going to have a film fest at the end of January. So we're very excited about this 2-day international film fest. There are flyers around. There's a flyer on the podium right there. So if you're interested in that, check it out. And then we'll start our Science on Tap series again in February. Those Science on Taps happen once a month and they vary on what day and they vary on topics. So if you're interested in getting information about those, there is a sign up sheet on the podium that you can put your email on, and it just, the listserv just sends out information about public events. So that is the only reason that you would get an email from me. The other way you can get information is if you go to HMSC's homepage, scroll to the bottom, there's a calendar of events there, and all the login information for any event that we might have will be available to you there. So a couple ways you can get information. For tonight, we are recording this event. Yes, Leigh? I forgot to ask her. And so I just pushed record and I figured I'd ask. And so that means that this event will be posted on the past seminar page in a couple of days. So you're welcome to watch it again if you missed something. If you want to watch the videos again, there's some cool stuff. Or if you want to share it with somebody who was unable to be here, feel free to do that. But I wanted to hand this off pretty quick to our speaker, but I wanted to do a quick introduction, and that means I got to put my glasses on. All right. So tonight's speaker is Dr. Leigh Torres, and Dr. Leigh Torres works with Oregon Sea Grant Extension, is associate professor with the Department of Fisheries, Wildlife, Conservation Sciences, and leads the GEM lab here with the Marine Mammal Institute at Hatfield. She has a bachelor's of arts from the American University in Washington, D.C., a master's in coastal environmental management, and a PhD in marine ecology and environment, both from Duke University. Leigh is a marine ecologist interested in understanding how marine mammals, seabirds, and sharks use their environment in context of behavior, space, and time. Much of her work is dedicated towards improving conservation management of protected species or threatened species. Leigh works everywhere, all over the place. She works in the Southern Ocean, she works in the subarctic, she works coastal waters of New Zealand, but tonight she's going to talk to us about her work here in Oregon. So we're excited about that. So I'm going to hand this off to Leigh. Leigh, you want to turn your mic on?
Thanks, Cinnamon. And this is good to go. Great. Thank you, Cinnamon. And thank you, everybody, for being here. I am so grateful that you came out in the stormy night and that you're here tonight, humbled by your presence. But I know most of you are probably here just to hear about the whales that swim off our coast. So we'll jump right into things. Right, we will jump. Do I need to turn it on? Click once here. Here we go. Okay, now we're ready to go. Okay, so most of the data and work that I'm going to talk to you about tonight is from a project that we call GRANITE that's been running since 2016 to 2022. And GRANITE stands for the Gray Whale Response to Ambient Noise Informed by Technology and Ecology. So this is a multidisciplinary project where essentially we're trying to find out what stressors, so multiple stressors, including ocean noise, but lots of other things, how that impacts the health and ecology of these gray whales that swim off our Oregon coast. But in order to do this, it's a very complex project. We have to collect a lot of data on a lot of different topics and understand it before we can actually get to the end goal of understanding what their physiological response is. So we're not quite there yet, probably another couple of years, but we have learned a lot along the way, along this journey, which is what I'm going to talk about tonight. But to start, I want to acknowledge all the funders that have helped us collect this data and do this work, including the Office of Naval Research, the Department of Energy, Oregon Sea Grant, NOAA Fisheries, the OSU Marine Mammal Institute, and the OSU Agricultural Research Foundation. I also want to give a big shout out to the fact that there is no way I do all this work by myself. I am surrounded by an amazing team who I want to give a big acknowledgement to, and you'll be seeing their faces throughout my talk because they're key to a lot of the aspects of the work that I'm going to be telling you about. So I'm going to introduce them. First, we have Casey Bierlich, who is my postdoc, and he's in the house. So give a wave, Casey. And then, unfortunately, my other postdoc, Alejandro Fernandez Ajo, he's not with us tonight. He's off doing some very important work at another location. But we do have in the house my PhD student, Clara Bird. So give a wave. And my other PhD student, Lisa Hildebrand. And then Kate Colson is not with us tonight, but she is probably online. So she's my master's student and also a big contributor to the project. So I want to give a big thank you to all of them. And yeah, we'll be seeing them again in the talk. So like all great things, it's better when you do it in partnership. And this work is like that as well. We are by far not the only people to study the PCFG gray whales. And this work, people have studied them for over 30 years. And many of the researchers that have been studying them for so long are part of the PCFG consortium. And one great thing that they have done over this long time period is taken photographs of these whales. And that allows us to build up a photo ID catalog. And essentially we can estimate age from that, because if a whale was seen 30 years ago, we now know that that whale is minimum 30 years old. And as you'll see as we go through the talk, that age information is really important to us, as well as sex information comes from either our fecal samples or genetic analysis. And a lot of that is all supported by NOAA Fisheries and curated by the Cascadia Research Collective. So I want to give a big shout out to these groups and all the contributors that help us collect and understand the data that we have. And I also want to thank you all. So this giving this talk here tonight is actually a big deal for me because these are our gray whales. And I really am excited to share all this with you all and to thank you because there's so many people in the audience tonight that have really supported me. Don't cut. Have supported me in so many ways to collect this data, whether it's childcare at a last minute, a piece of gear that I suddenly need, a good piece of advice, a good conversation that gives me an aha moment. There's so many different ways, a good soccer game to burn some stress. So I'm really grateful that I get to share all this amazing work that you guys have helped contribute to with you guys. Okay, so let's get into it. Who are the PCFG whales? Well, as many of us know, there is the Eastern North Pacific gray whale population, which is most of the gray whales in the Eastern North Pacific. And all gray whales in the Eastern North Pacific, they spend the wintering time in the lagoons of Baja, Mexico. And when that's over, most of the population migrates. So about 20,000 individuals in the Eastern North Pacific population, they migrate up to the foraging grounds in the Bering or Chukchi Sea in the Arctic. But there's this strange, and up there, they feed mainly on benthic amphipods. So gray whales dive down to the benthos and they sort of scrape the seafloor and suck up amphipods. But there's this strange group of about 250 individuals that don't make this full migration all the way up to the Arctic, but instead truncate that and spend their summer feeding period, which is about May to October, in the waters between Northern California and British Columbia. And these are called the Pacific Coast Feeding Group, or the PCFG whales. And they feed in this shallow nearshore water. So many of you that live here in Oregon have probably seen them at some point. So there's some really basic questions about this PCFG population, which I hope to get closer to addressing here tonight with you all. We're nowhere near knowing the full answers to these, but we're scraping closer, ever closer. So why do these whales use this range instead of going to the Arctic? That's one basic question. How do they feed and what do they eat? How does this different life history strategy, instead of going to the Arctic, how does it affect this PCFG gray whale population? And what risks do they face? And how do they cope with those risks? So that's some of what we're going to try to talk about, or learn about. So, for starters, I'm going to tell you about how we collect our data. So I'm going to take you on a journey with us for a day in the field. So it all starts by loading up our trusty research vessel, Ruby. So many people might have seen this in the parking lot right out here on good calm mornings where we load all the important gear. And this is a undervalued step in the process, because if we forget one critical thing, it throws off the whole day. So then we launch right here at the South Beach Marina, and then we go out on survey and we look for whales. And hopefully soon thereafter, we'll spot a blow. At which point we slowly go over to the whale, and the first thing we do is conduct photo ID, basically taking pictures of the whale's left side and right side, and fluke if they fluke up. And this very traditional basic form of data collection is so essential to us, because it really is the glue that links all of our data sets together, because everything we do is linked back to that individual. And again, here from those photos, we can know the age of the animal, either whether it was first seen as a calf, we can know its exact age, or its minimum age based on when it was first ever photo ID'd by ourselves or this collective group of people that study the PCFG group of gray whales. So we also fly drones over the whales. And as you can see here, we launch it right from the side of the boat. And there we have Lisa grabbing the drone out of the air as Clara carefully pilots it down to her. And with this data, we are either doing photogrammetry, trying to measure the length of the whales and the body condition of the whales, and looking at the behavior of the animals in the water. So one important thing that we do is collect fecal samples from these whales, and this is very opportunistic. It's just based on when we see them poop. It's a very exciting moment, poop. And we carefully drive over behind the whale, making sure the whale moves out of the area, and then we just get out our nets and swish through the water and try to collect as much fecal sample as we can. And you can see here these big smiles on my team when we collect a good sample. And that's because we view these fecal samples as biological gold, and they really are. We can do so much with these fecal samples. And we can look at the whale's genetics, the prey of the genetics, the microbiomes, the microplastic loads, and the hormones. So hormones are the major thing that we're interested in, and I'll be talking about later. We can collect it non-invasively, which means we can collect it multiple times from individuals throughout a season and between seasons to understand how those metrics might change throughout their life. So we also assess the prey and the habitat. So here this is one way we assess prey, is by basically the small net sampling device off that downrigger, but it can be very effective when we collect prey. We also drop GoPros in. Basically, it's a GoPro on a stick with a weight that we drop on a line off the side of the boat right where we're with whales. And it gives us this great underwater view of what the habitat is like there where the whale's feeding, what the prey might be like. So here you can see all those little bugs in the water. Those are essentially the mysid zooplankton that the gray whales are feeding on. So these are two methods that we use to assess the habitat available to the whales and what they use. In the past 2 years, we've been doing this suction cup accelerometry tag deployments. And here's a video of us deploying one of these tags. So that sound you hear is the camera shutter. So this is a GoPro on Lisa's helmet, and she's taking pictures of the whale. This is our collaborator, Dave Cade, on the bow. So yeah, it's a very exciting. So it's a tricky thing. So I'm driving the boat and you can see that the whale swims away. It's tricky to get right up there. And it's also very exciting when we get it done right. But it's equally exciting when we get the tags back, because these are expensive tags, but the data is on those tags. So we really need to get them off. So they come off eventually, 30 minutes to 24 hours later, and we have to go find them. They're floating around, and there's some adventure stories there about finding those tags, but they're super valuable and they give us great data that I will tell you about later. So throughout this whole time period, we also have hydrophones deployed in our study area. We have 2 of them deployed, and they measure the soundscape, so what the noise is like in this underwater environment. So in late May, we deploy our hydrophones there, and then we go back 5 months later and pick them up, hopefully, and then we have them on deck, and those hydrophones are full of data that tell us what the whales were hearing throughout that period. So this is our study area. And you can see where we deploy our hydrophones here, one by the Otter Rock Marine Reserve. Oh, no mouse. Oh, there it is. Okay. So up there is where we would deploy one. And then we have this other one by the Newport Jetty here. So a sort of a quiet soundscape and a louder soundscape is what we expect and why we put our hydrophones there. So the study area is very coastal. It's our nearshore rocky environment. And well, it's a mixed habitat of rocky reef as well as sand and mud in some places. So this is some plots of our sightings across 6 years of our study. I haven't yet plotted 2022 yet, but you can see that we have sightings across our whole study area in each year, and quite a high number of sightings. And that's a testament to the individual whales' residency and fidelity to this area. And so that's really something you'll hear me talk a lot about tonight, is the fact that these whales are whales we know, and they return to this area year after year, and with high re-sight rates and high site fidelity. Site fidelity meaning they come back to generally the same area over time. And to show you exactly what level of site fidelity, I'll walk you through this graph here, where you have on the x-axis on the bottom are our individual whale names. And then on the y-axis here, we have year. And each box has a number and a color, which means how many times we saw it in that year. So you can see up in the top left there, we saw this individual Soleil in 2021 17 times. And in fact, we've seen that whale, it's a female, every year of our project. And so this is just for the whales that we've seen 10 times or more throughout our study period. And so really it's powerful data that allows us to understand how these animals might change their behavior or body condition or hormone levels over time based on other factors. Okay, so we're going to jump into some results now, starting with our behavior analysis that's coming out of our drone footage that's led by my PhD student, Clara Bird up there. And so Clara has painstakingly annotated 6 years of drone data that we've collected, which is, I don't know, she could tell us over 300 hours, basically marking every behavior that these whales do. And so our research questions coming out of this are: what foraging tactics do they use? Is there specialization among the individual whales? And are these patterns of tactic use related to either their age or their sex or their size? So there are a number of different behaviors that we've seen, I think over 45, but we're concentrate on the foraging tactics, and these are 4 of the main foraging tactics that we've filmed on multiple times, quite common. And one is upside down swimming, which is what it sounds like. They swim upside down to feed. The other one is side swimming, which in this case is stationary side swimming. And here it's really amazing. They use their pectoral fins so much to stay stationary. They're basically sculling to hold themselves in place and feed. Then we have headstanding, which again is what it sounds like. The whale's head down with its fluke up, and they can headstand sometimes in sediment or in rocky kelp areas. And then they have side swimming forward, which again is what it sounds like. They swim forward and often have a jaw snap there at the end to grab what they're after. So Clara, the results I'll show you, these are initial results. So we're still working on it. We got another year of data to add into the mix. But these are some of our initial results. And what we want to know basically is, are there clusters of behaviors, right? Are certain whales performing certain foraging tactics more often than they might otherwise by random? And so this is the results of a clustering analysis. Yeah, okay, so you can see that. So what we see is that in fact there is certain groups that perform different foraging tactics. So you can see, if I can get my mouse, there we go. This group here generally performs this side swim stationary more than others, whereas this group here is in this side swim forward. So in fact, these were significant differences in significant clusters of behaviors, which shows that certain whales certainly do certain behaviors more than others. So the next question was why? What are the patterns driving those clusters of behaviors? And that comes through a little bit. Again, these are initial results, but it's pretty exciting to see. And here we have on the x-axis those same behavior states on the bottom, so side swimming and head standing. On the y-axis, we have the total length of the whale. And then each one of those dots represents a whale, and it's color-coded by its age. So the lighter the color, the younger the whale. And what we see here is that the younger, shorter whales tend to not do the stationary feeding behaviors, right? So this group here seems, oh, no, I can't point. This group here, the side swim forward, are the shortest ones and the youngest animals. And what we think is that it takes a lot of energy and learning behavior to be able to master those stationary behaviors. And in fact, we saw this really cool, well, we filmed it this summer where we filmed a mother and a calf, where the mother was doing this headstanding in a rocky reef environment, very cool behavior. But meanwhile, its calf was right there. And Clara and I were cracking up in the boat because the whale was literally doing barrel rolls, and it was hysterical. And we were like, wow, it's just playing, look at the funny whale. But then I watched it several times that night, and I actually think the whale was trying to mimic its mother and learn. It was trying to go down and do headstands, but its pectoral fins were just glued to its side. It hadn't figured out, I think, how to actually turn and get its head down. So anyway, that's just anecdotal, but I think we're onto something here with where these whales need to learn how to master these behaviors, and that takes time and strength. So stay tuned as Clara continues through with her PhD, and we'll give you the results, the final results later. Okay, but the other area that we're getting behavior data is coming from our tag analysis, which you saw a clip of us deploying those tags. And here this work is led by my master student, Kate Colson. And what you're looking at here is a photo taken by a whale. So this is a tag on the whale's back, and you can see the lighthouse there. Isn't that cool? So the research questions here are, what behaviors can be detected by these tags, and what is the energetic cost of those behaviors? So here are just a couple of cool clips. Again, this is filmed from the whale's back. So this is one of those headstanding in kelp behaviors. So the whale is head down here, and you can see it kind of raise its head every now and then as it takes a mouthful. Yeah, there. And so that's the kelp nearby, and you're going to see some sediment there, a sediment plume come up from the side, which is probably just sand around the bottom of the kelp as it moves around. So it's really great to get this underwater perspective about what the whale is seeing and how it's feeding, to link up with what we're seeing from either the drone or the boat. And then this clip is just super cool. So here we have the whale, the tag on one whale, but it's got a friend with it here that's feeding right alongside of it. And if you keep your eye on the friend, as it'll come back into view here, as they swim through this kelp and feed, and you can see mysids going by, but you're actually going to see this whale do something really cool. Right there. Did you see it? As it just opens its mouth, takes a big gobble. And it's really cool to see that, because these gray whales really traditionally everybody thinks they're just benthic feeders, but they're obviously doing another foraging strategy as well, feeding more pelagically, going through the water, opening its mouth. So yeah, really cool to see this. Okay, so in addition to the cool video that we get out of these suction cup tags, we also get accelerometry data. So this is like what's in your phone, right? Which, you know when your phone knows when it's up or down or side or rolling over. It's the same sort of thing on these tags. We have data in the X and Y and Z axis. And one thing we can get out of these tags is the depth information. So here you can see the tag will tell us when it's at the surface, and that's the whale breathing here, and then it dives down to about 12 meters and does some stuff, and then goes back up for another breathing event, surfacing. Another axis of data that we get is about the roll of the animal. So here, when the whale is at 0, the whale is, flat at the surface. But then you can see that this whale consistently rolls to about 115 degrees, and that's just past 90, right? So definitely a roll event. And you can see in this is data from our tags, that the whale goes into this roll event, briefly out, back into a roll event, and then back out again. Another axis of data that we get out of the tags is the fluke stroke. So that's what the little green line going up and down can represent how the whale is fluking. And it's from that fluking information that we will eventually get to the energetic cost of these different foraging tactics. As we're learning is that some of these tactics cost more energy than others. Okay, so when we put all of these axes together, these data, we can actually recreate the track line of the whale. And so in this spaghetti thing here, I'm gonna press play here in a second, and you're gonna see a whale swim around. The blue is when the whale is at the surface, and the yellow is when it's underwater. And so you can see here that the whale basically, rolls on its side quite frequently, comes up to the surface, does a tight turn, and then rolls back on its side as it's sort of plowing through the bottom and feeding. And then, so this is what it does time after time, these tight circle, and then here's a long roll event on its side while it's feeding and coming back up. So it's really powerful information to help us understand, again, what these whales are doing underwater. Because when they're at the surface, they're only at the surface for a brief moment of their life. So it's really helpful to get underwater with them, ride on their back for a bit to figure out what they're doing underwater. Okay, so now that we've talked about their behavior, let's talk a little bit about what they're actually eating. And here this work is led by my PhD student, Lisa, in the back. Thank you, Lisa. But a lot of this work was actually part of her master's work that has already been published. But we rely on it quite a bit. So our basic questions here were, what species of do these gray whales eat, and what is the caloric quality of these of their prey? Okay, so as I showed you, we collect zooplankton while we're out in the field. And when we go through and identify what is in those that catch, it basically boils down to these 6 main prey species. 2 species of amphipods, 2 species of mysids, and 2 species of crab larvae. And these species are similar to what's found in other areas where the PCFG feed across their range. So other people have documented the same prey species. And so what we wanted to do, we wanted to know whether the caloric quality of these prey that the PCFG whales feed on is how that compares to the caloric quality of the prey in the Arctic that 99.9% of the gray whale population feeds on. So we went to the literature, and this is the main prey comprises about 70% of the prey that the ENP feeds on in the Bering and Chukchi Sea. And when we looked at the literature, it said that the caloric quality, the caloric values of this amphipod is about 2 kilojoules per gram. So Lisa painstakingly went through and calculated the caloric value of these critters. And in fact, these 3 had lower caloric value than the amphipod in the Arctic. But this crab larva, the Dungeness crab megalope, had higher, in fact, twice as high caloric value than the amphipods in the Arctic. So that was really interesting to us, but many of you, especially in this audience, would probably know that Dungeness crab larvae are not constantly available to these gray whales. They come in pulsed availability into the ecosystem. And so, but the mysid shrimp are available, throughout the gray whale feeding season in this area. And so we were interested in how their caloric value compares. And in fact, one of them, the Homeomysis sculpta, was a little bit lower, but the Neomysis rea, this one at the bottom here, had higher caloric value. So this was really interesting to us because what it lent us to believe is that the prey here has equivalent caloric value as the primary prey of the ENP that goes all the way up to the Arctic. So the question remains is why would most of the population still go up there to feed when they could feed here on higher or similar caloric value? So, but now we wanted to know was how much prey does a gray whale need to eat if it could, now that we know the caloric value of these prey. And so what we did is we went to the literature, and we, this paper by Villegas here, they calculated the energetic requirements of a gray whale, of a lactating or a pregnant gray whale on a daily basis. And then we extrapolated the caloric values that we had just calculated for our prey. And here, I'll go to the mouse to explain. Okay, so here, if a whale could just eat its whole time it's here, this Dungeness crab megalope, it would need less than 1 ton of prey a day. But as we just discussed, that's probably not realistic, nor is eating Neomysis rhea all the time. So we calculated this composite prey, which is 1/3 of the 3 main prey species here, the amphipod and the 2 species of mysid. But when we compared that to what was available to the whales feeding in the Arctic, these amphipods, you can see that it's quite similar. So both populations of whales, the ones up in the Arctic as well as the ones feeding off our coast here, basically need to eat about, pregnant or lactating females, 1.5 to 2 tons of prey a day to meet their energetic prey demands. Okay, so now we've talked about what they're eating. Now we're going to talk about what comes out the other end. So hormones are the main thing that we do with the fecal samples that we collect. And this work is being led by my postdoc, Alejandro Fernandez Ajo. And our main research questions here are: what are the patterns of reproductive hormones in these whales? How does it vary over time and individual? And what causes stress hormones to vary? And I'll just give you a little taster here of what we're finding. So this work is really new. And so a lot of what we've been doing to date is just establishing the baselines of how these hormones vary by different demographic unit and so forth. So in this figure here, you can see, hopefully you can see, this is testosterone, so the male reproductive hormone on the y-axis. And on the x-axis, we have different demographic units. And what you should take away from this is that those mature males have very, very high testosterone levels sometimes, much higher than the other groups. And this is, normal. When mammals go into reproductive states, their testosterone levels increase. So this is just a basic result, but a very important one to help us understand how testosterone will vary over time in individuals. So another really important metric that we're using to understand the physiology of these whales is body condition. And we're doing this through our drone data. And here this work is led by Casey Bierlich, who's in the audience. So what our main questions here are: how does PCFG body condition change, either by year or time unit or by demographic unit? And so what we want when we collect our data is to get that perfect image of a whale flat and straight at the surface, like this ideal image here. And then we bring it into our software and we measure it. So we measure the total length from nose to tip of the fluke there, as well as the widths of the animal at different percentages of body length along. And so this area that's in green, in that green box, from 20% to 70% body length, we then run through a really awesome model that Casey's created to account for any uncertainty in our measurements. But essentially what we get at on the other end is a really important metric of body condition that we call the body area index, so BAI. And what that is, it's similar to BMI in humans. It's a length standardized unit that we can, of body condition, that we can use to compare their body condition to another individual that might have a different length or be in a different demographic unit. So it's essentially the surface area of that part of the body between the 20% and 70% body length divided by its length. So here's just a very simple example of kind of the data that we get out. We have body area index on the y-axis there and the demographic unit on the x-axis. And what you can see here is that those calves, the chubby calves and the pregnant moms have the biggest body area index, large body condition, whereas the lactating females are the skinniest. And that's because they are shunting a lot of their energy, their reserves and their blubber to their calves. So we can link these two physiological metrics together, so body condition and stress hormones, to look at how the two co-vary. And this is a really interesting result that we've produced, where we can see that skinny whales have higher stress levels than fatter whales. And so here you can see that the whales, the points on the left part of the axis are the skinny whales, but they have the highest cortisol levels, the highest stress levels, whereas the fat whales have the lower cortisol levels, lower stress levels. So this is a really important fact for us, and it helps us understand, okay, well, we need to account for the whale's body condition if we're going to try to understand how they might have a stress response to some disturbance event. So we've also been able to document high variability of the body condition of these whales over time. So as I've said, we've been studying this whale, doing these same methods since 2016. And you can see that at a population level, the body condition of these whales has gone up and down over time, including in the last 2 years, not including this most recent year. We haven't quite finished processing that data, but the 2 prior years were pretty low body condition for these gray whales. And we think that this probably has something to do, a lot to do, with the environmental conditions that control their prey availability. So there has been what's called an unusual mortality event of gray whales since 2019. And due to the rise of stranded gray whales on our beaches between Mexico and all the way up to Alaska. And many of these gray whales have been emaciated. So an unusual mortality event is something NOAA declares when there's a significant increase in dead animals of some species or another. And so this has been going on for gray whales since 2019, with many of the animals being found in a skinny body condition. So one of the main questions that we had in my lab was, is this UME impacting the ENP population? So those whales that go all the way up to the Alaskan Arctic to feed, or the PCFG, or both equally? And so in order to address this question, we compiled our drone data with people that were collecting using the same methods, used flying drones over whales in the lagoons in Baja, or during that same period between 2017 and 2019, so that 3-year period leading up to when the UME was declared, that unusual mortality event. And so we have our drone data collected here in Oregon, and then we compared it to the drone data over whales looking at their body condition in the lagoons. And here we do make the assumption that all of those whales that they imaged in the lagoons are of the ENP population, just because the sheer number, there's 20,000 of them and only 250 of the PCFG. So it's most likely that they were imaging whales from the ENP population. So our hypothesis here with this work is, if there are similar environmental conditions regulating the prey availability on the 2 foraging grounds, here off Oregon as well as in the Alaskan Arctic, we would expect the body condition of both the ENP whales and the PCFG whales to decline, together at the same rate from 2017 to 2019. But what we found was not that. So we actually reject the hypothesis. There it is. It's rejected now. So what we see here is that while the brown line represents the ENP population, their body condition at the different time points that we could monitor them between 2017 and 2019. And in fact, overall, their body condition did decline during that period leading up to the UME. But in contrast, the PCFG population, which is that green line there, you can see that over time, they generally actually increased in body condition. But what's really interesting and caught our attention is the fact that mostly the PCFG population has a lower body condition. They're skinnier than the ENP population. In fact, they only matched their body condition at that last time point when the ENP population was at their skinniest and about to go into this UME period. So this led us to this question, well, are ENP whales inherently fatter than PCFG whales? So to answer this question, we collected an we compiled, we didn't collect it ourselves, our collaborators did, but we compiled an opportunistic data set of aerial images of gray whales feeding in the Alaskan Arctic. And we compared the body condition of those whales to our whales. And in fact, it's true. It does seem so that the ENP whales feeding in the Alaskan Arctic are fatter than the PCFG whales here. And so this was quite striking to us. And so we, as good scientists, dug a bit further. So again, led by Casey here we're looking more closely at the body morphology comparison between our gray whales here, the PCFG whales, and those ENP whales in the that feed in the Arctic. And here our questions are PCFG and ENP whales the same length? And do they have the same morphology? So to answer this question, we used, again, our data set here that we collected by the GEM Lab, our drone photogrammetry. The age comes from our photo ID data, and then sex either comes from the fact that we saw a mom with a calf or from genetics. And what we did was we built these growth models where you have to know the age, sex, and length of the individuals. And so to do this for the ENP whales, we actually used a data set that was compiled between 1959 and 1969 through scientific whaling. So this was a dedicated effort off the California coast where they actually killed whales and did a lot of scientific measurements, which are all published and available. So we used that information. So the length of those animals were measured manually. The age of those animals was determined through earplugs. So it's like long, waxy earplugs where you can look at the number of rings, like you count the number of rings in a tree ring. You can do similar to where each layer is a year of a whale's growth. And then sex was determined by their by examination. So Casey built these very, very interesting growth models. So I'll walk you through these. So here we have a growth model for the females of the ENP and the PCFG. So the ENP is in blue and the PCFG in green. And then on the right, we have the same growth curves for males. And what we can see for both females and males is quite striking, where the growth for females, you can see that they grow slower. So we have age on the x-axis and length on the y-axis. So as the whale ages, we can see that those PCFG whales are growing slower and their asymptotic length, so their maximum length that they reach, is about a ½ meter shorter for females and a full meter shorter for males. We were pretty wowed by that. That's pretty amazing. That's a significant difference. And so this leads to a lot of really interesting follow-up questions, which we are currently digging into. So one of which we're looking, okay, so they're shorter, but is their morphology actually different? And to do this, we use the same data set, so that scientific whaling data set. They measured these measurements, the snout to the blowhole. They did that measurement as well as the fluke span. And we realized we could do those same measurements in our drone data. So we compared these. And in fact, we see a similar pattern here where the PCFG whales have smaller skulls. So that's that top plot there, again, broken out by males and females. And then fluke span, which doesn't look like quite as much separation, but that's equally statistically significant. So these PCFG whales do seem to have smaller skulls and smaller fluke spans than the ENP whales. So this is pretty interesting and raises a lot of questions. So why would the PCFG whales be smaller? Is it perhaps an adaptive, morphology to their, strange feeding behaviors here in the shallow, reefy environments? Maybe they don't need to get as big because they have a shorter migration, and so they don't need as much energy stores. But equally important to consider, we think, is what are the consequences of this smaller body size? Perhaps because they have less energy stores, they might be less resilient to disturbance. Or perhaps they might have a lower reproductive capacity because they don't have as much energy stored to have as many calves as frequently. So these are some really important questions to consider. And for this last one about reproductive capacity, I'm just going to take you back to thinking about our hormone data for a second, because what we're trying to do here is develop a non-invasive pregnancy test to try and help us understand those reproductive rates for these animals. So here what you can see is that we have detected a difference in the progesterone levels. So that's the female reproductive hormone. And that's what that log P4 there is on the y-axis. For pregnant females, it does appear to be higher than for non-pregnant females. And equally interesting is the fact that these whales are fatter. Pregnant females are fatter than non-pregnant females, so just like humans. So what you see here in this plot is, again, the 20 to 70% width, length along the whale, and the red are juvenile females, the green are lactating females, And then you can see here this high curve here, the blue, that's the pregnant ones who have significantly higher widths, especially at this 50% body length, which is this group here. So we can combine these two metrics, these two non-invasive metrics of body condition, so body width, and hormones to determine pregnancy. And so we're really excited about what this will allow us to do in order to determine or assess, hopefully estimate the pregnancy rate of this population, as well as the pregnancy loss rate. So we're working on it, and come back hopefully in a year or so and we'll have the answer. Okay, so we've been talking this whole time about what we've been learning in terms of their biology and ecology, but now I want to shift a little bit and talk about, you know, the stressors that this population faces because of their anthropogenic habitat that they live in right off our coast here, as well as the consequences to this group of rapid environmental change. So I'm going to start by talking about our work looking at the microplastic consumption of this population. And this is a project that we affectionately call Zoop to Poop. And that's because we look at the microplastic loads in the zooplankton that they eat, as well as the poop that comes out the other end. And so the main research questions with this is what kind of microplastics are the whales consuming? What is in the zoop and in the poop? How much microplastic is in zooplankton prey, and how much microplastic does a gray whale eat in a day? So here what we see is just comparing what was in the zooplankton, the microplastics in the zooplankton versus the microplastics in the poop. And the first thing that I want to point out is that there were a lot more fibers. So these are small little fibers in the zooplankton than they were in the poop. The poop had a lot of bigger things, these pellets and fragments and film than what was found in the zooplankton. And the microplastic found in the whale poop was much larger. And so this schematic hopefully helps explain what we think is going on. So while the mysids, sorry, not the mysids, but the zooplankton, which includes mysids, probably feed on the the smaller microplastics like the fibers. The whales do eat that, but in addition to that, because they're benthic feeders, they're eating ambient larger microplastics that fall through the water column. So these are heavier things that don't just drift in the water column, but instead settle on the sea floor there. And so when the whales are feeding benthically, they're ingesting both the fibers that are in the water column, as well as in their zooplankton prey, as well as the larger microplastics that are just on the sea floor. So then when we collect their poop in our net, we're collecting both the larger microplastics that the whale has ingested, as well as those fibers from the zooplankton. So we calculated the number of microplastics per each of these three main prey species of these gray whales, and it turns out that it's about 4 microplastics per per gram of these prey items. So what we wanted to know then was, well, then how much microplastic does a gray whale eat in a day? And so this plot should hopefully look a little bit familiar to you, because it's again showing how much, how the number of individuals of each of these prey items a whale would need to eat to meet its daily energetic demands, right? And so what we did was we extrapolated our recent results that showed us the number of microparticles per individual prey items for our 3 different prey species, as well as that composite prey. And then we extrapolated to how many individual prey items would the whale need to eat in order to meet its energetic demands for a day. And so what you can see here is that for a lactating gray whale, if it only ate the most energetically rich Neomysis rayi, it would eat about 6.5 million microparticles per day to meet its energetic demands. And then for a lactating female, if it ate that composite prey, it would ingest about 13 million microparticles per day. So these are large, scary numbers, and we are quite astounded by them. And really what it drives home to me is that we really need to start understanding what the impacts of this ingestion are on the health and physiology and growth and reproductive rates of these animals, because they certainly are eating a lot of microparticles every day. Okay, so now I'm going to shift and talk about our noise, our soundscape results. So whales, like all cetaceans, really rely on sound. It is their main sensory organism that they use to find food, find mates, migrate, communicate with each other. So our main questions here were, what is the soundscape like in this region? So you can see, again, I've put this map up that show where our hydrophones were, one by the Otter Rock Marine Reserve up north, and then one right off the jetty here, off the port, Newport port there. And how does the soundscape vary over this time and space? And then what are the impacts of this variable soundscape on the whales? And so just to show you, or yes, you'll have a listen here to a natural soundscape. So that's very pleasant. That was recorded up here off the Otter Rock Marine Reserve. And I'm going to contrast that with an anthropogenic soundscape. So be ready. So this is the sound of a boat going right by. And you can kind of actually also hear the Depot Bay bell buoy going in the background. So quite different. So you can tell right away that there's a big difference between a natural soundscape without the influence of vessel traffic and one with vessel traffic. So this is our data from 3 different years. So we have 2017, 18, and 19. On the left is a spectrogram. These are all spectrograms from the Newport area, the Newport hydrophone, in contrast to the spectrograms from the hydrophone up by that marine reserve by Otter Rock. And so each one of these little boxes represents an hour of time. And on each day of each hour of the year for each day that the hydrophone was in the water during these periods. And the color of the box is color coded by the decibel. So this is essentially how loud that noise is, with the brighter colors, so the reds and the oranges and the yellows being louder. And so you should be able to immediately see that it's noisier, louder by the Newport, but at the hydrophone right off the Newport jetty there, in comparison to by the Otter Rock Marine Reserve. But what was also really interesting to us is that diel pattern. So right at 7 a.m., there is a huge increase in the noise. So you can see there, it goes from being fairly quiet blue to very bright. And that's because all the recreational and charter boats and fishing boats and whale watching boats, everybody, they're leaving the harbor at about that same time. And then that noise seems to die down around 3, 4, 5 in the afternoon when those boats come back into port. So what we see here is our soundscape here is highly vessel dominated with diel pattern. So what we wanted to know was, what are the impacts of that? And so we did some modeling comparing those cortisol levels from the fecal samples to the number of boats on the water. So we looked at the number of boats on the water relative to those stress cortisol levels the day that we collected the fecal sample, the day before, 2 days before, 3 days before, 4 days before, a cumulative, all of those, because we wanted to know what was really impacting the stress levels. And as it turns out, it is the number of vessels on the water the day before. And that is because that is the gut passage time. So a whale has to eat food, process the food, it secretes the hormone into its digestive tract, and then comes out the other end. And that whole process takes about 24 hours. And so we do see a really significant correlation here between their stress levels and the number of boats on the water. And that's because there are a lot of boats on the water. So when we're out on our boat, so these are just photos that we've taken, we often see boats in close contact with the whales, whether intentionally whale watching or unintentionally. But these whales live very close to shore. This is where they're feeding all the time, and their boats going through their habitat all the time. So, and there are impacts of this. So whether the impact is just the sound, is the sound really what's impacting the whales, or is it the presence of the boat and the risk of collision? And so what we have seen is a number of boat strike events. And so I have a few examples here that I just want to point out were all documented just this past year. So this is a whale boxing that we saw with its nice pointy knuckle there on the left. And 5 days later, it had this chop on that knuckle there. That was quite disturbing to see. Here's another example of a whale this summer that we documented before. You can see no injury, and then after, it's a clear propeller strike there. And then finally, here's another example of this individual that we call burned, where the injury is actually on its rostrum there. So you can see after that big divot there, whereas before it didn't have it. So this is a strike right to its head. So we are very concerned about what the impacts, carrying these injuries might have to them, both in terms of an infection, the energy it will take for them to heal from that, the missed reproductive opportunities. So all of that has an impact on the individual as well as the population's health and dynamics over time. And of course, these oh, sorry, just one more example. And this one's actually a very cool one, because this is sad for equal, but it's cool for us, because we saw this whale on the 19th of June and it was totally, happy, fine. But then we saw it the next day on the 20th of June, and it had a clear, fresh propeller strike. And what's interesting is that we had collected 2 previous fecal samples from this individual where, it had pretty low cortisol levels, but we were managed to collect a fecal sample from the same individual within 24 hours of that vessel propeller strike. And you can see that that cortisol level is 3 times as high. So again, we're seeing that our hormone data is managing to capture these stress events, and that these vessel strikes are very stressful to these animals. So, but we know the vessels aren't typically out there for just a joyride. Many of them are out there fishing, and that fishing also has impacts on the individual. So again, you can see in just these 2 photos in the bottom right there, that the whales feed right around crabbing gear all the time. And we see that, and we're always a little bit nervous about that, because we also see the evidence of entanglement wounds on these 2 images on the left, as well as this summer we documented a whale actually entangled in monofilament line. So this happens, right here off our coast, and all of that again has these, immediate physical impact to the animals, but it can also have long-term impacts to their health. And here's an example of an individual roller skate where we documented the whale with a nice pretty fluke in 2018. In 2019, it had a wrap around its fluke there. You can kind of see its fleshy fluke. And then after in 2020, it clearly had a very damaged fluke. And it was trying to feed right off Otter Rock, but it was not feeding, not diving very effectively. And sadly, we haven't seen this whale in the past 2 years. So we are, sadly nervous about it. So these non-lethal impacts to these animals can lead to, death or lower reproductive rates. Oh, yeah, so there, I circled it for you. Okay, so the last impact that I want to talk about tonight is environmental change. So, and this is likely maybe the biggest thing these animals are dealing with, and animals across our planet really is the effects of climate change. So through our work, we've documented these significant relationships between the zooplankton, the kelp habitats where these gray whales live, and the gray whales feeding. And so we have monitored the same 12 sites for 6 years, where we looked at the amount of kelp, urchins, zooplankton, and whales at these same 12 sites. And this is images from our sites in 2016, which you can see have, great kelp, very healthy habitat. But by 2018, these same exact locations look like this. Lots of urchins, the kelp basically chewed to bits by the urchins. And so we were really interested, okay, well, we know that we can see that kelp habitat is very important to whales. So can we detect those changes in our data? So this is the typical sort of reef ecosystem in the Northwest, where we have, a predator, in our case, sea stars, because we don't have sea otters on our coast here, but they control the population of urchins, so they don't overgraze on kelps. And then the kelps provide great habitat for reef biodiversity. But there has been this well-documented trophic cascade where there was a sea star wasting disease. So that led to the sea urchin population being able to increase dramatically. And so they overgrazed the kelp. And at the same time, there was this big warm blob event so warm water temperatures that basically caused the kelp not to be able to grow and provide great habitat anymore for that reef biodiversity. So this has been documented in Northern California, and we have hints of it here off our coast. But what we wanted to know was, is there an impact on the mysid zooplankton prey of the gray whales and the gray whales' behavior themselves? And so after a lot of modeling and great work by Lisa and team, these are some of the outputs of our results. And what you can see here is that over our 6 years, so from 2016 to 2021, you can see there was a dramatic increase in the presence of urchins. And at the same time, you can see a dramatic decline in the kelp presence, the kelp health in the area, the zooplankton abundance, and the numbers of gray whales feeding in these areas. So in fact, what we see is that these changes in habitat are impacting gray whale foraging and potentially their health, because as we know, the zooplankton that they feed on leads to their increased body condition. Okay, just a little bit more. We're at the end. So I've thrown at you a lot of our results about the PCFG gray whales. And so what I want to think about now is how these animals are really very distinct from that larger ENP population. We've learned that they are geographically distinct. They have high residency and site fidelity to this region here. They have distinct foraging strategies and prey. They have lower body condition and are smaller than those ENP whales. And importantly, they have distinct threats. So I want to highlight, this unique culture and life history of these gray whales that feed right off our coast here. And what I really want to point out is that currently the management of these gray whales is no different than the ENP. So they are not recognized as a separate stock or a separate subpopulation, and which would mean that they get management regulations particular to their needs. And so right now they are managed in no special way, despite the fact that they are facing distinct threats in this area from fishing gear, from vessel traffic, from changes in their habitat. And I do think that it's time that we collectively just begin to discuss how we can better protect them and allow them to live in this habitat that is quite anthropogenically influenced by our activities, so that they can survive and this distinct, cool, really interesting population can persist into the future for a long time to come. So that's all I have. I'll invite you to visit our website or blog or chat with me or my students in the house. And I really want to thank you all for coming out tonight and listening.
All right. Thank you so much, Leigh. That was amazing and impactful of a lot of information. So I am sure there are questions. So because we have two audiences, one here and one online, we'll take turns. And like I said, there are two mics for those that are in the room, mine and the one up there. So feel free to either move to that mic or raise your hand. But I'm going to start with the folks online. Do we have any questions?
Yes, there's several questions. First, from the mean poop content in using your calculations of particles consumed, can you roughly compute the proportion of particles that are retained by the whale?
No. No, but it would be great to do. So part of the problem is that when we collect the poop sample, we really don't know how much of the fecal plume we're actually collecting. And so like we could be collecting, all of it. And so we're collecting all the microparticles, or we could just be collecting, a small portion of it. One thing I meant to point out in that plot of how much microparticles they're consuming a day, which is scary, is the fact that that's actually a conservative estimate, because that is only calculated based on the amount of microparticles that they consume through their prey. We didn't, incorporate any estimates for how much they're consuming just by ambiently eating microparticles in the benthos.
All right, questions in the room. Do you want to stand? Turn the mic on. There's a little slider. Thanks, Tommy.
Do you know if once a PCFG whale, always a PCFG whale, or do they sometimes go to the Arctic? And then kind of a follow-up to that is an offspring of a PCFG whale destined to be the same?
Great question. In fact, we have that same burning question. We're working on that. I think my gut tells me that it's a spectrum. And in fact, we use this term these days called PCFG NIST. So we do have some whales that we see all the time, every time we go out, essentially. And that's a very PCFG whale. It probably doesn't go to the Arctic very much at all. But then there's some whales that we see once, twice. And it might be, because, well, first of all, the PCFG range is bigger than our study area. So it might have its special spot, up in Canada someplace. But then it might also be a whale that, goes up there and comes back. And so part of my PhD student Lisa's work is addressing that very question. And hopefully we'll get there. Your second question about calf recruitment, definitely there are many calves that we see of PCFG gray whale mothers that come back and recruit into the population, and we see them over and over again, and that's super exciting to see. But there's also whales that we see with calves that we haven't seen that calf again, and we don't know if it died or if it goes someplace else. But those are really important questions to answer about internal recruitment to the population and external recruitment, and so we really hope to get there with our colleagues.
All right, how about the next question online?
Do you think that the body morphology of the current ENP whales could be significantly different than the information in the data from the scientific whaling due to the time passed and changes in the environment after the data was collected?
Yeah, another great question. Yeah, so maybe the whales got smaller over time, and it's not that they're different. So the reason that we think that that's not quite the case is because there has been photogrammetry done of gray whales, the ENP gray whales, in actually 2000. There was some aerial photogrammetry, and down in the lagoons, that same data set that we used for that comparison in 2017, and those whales were longer also than the PCFG whales. So we do think that it's not that they've gotten smaller over time, it's that this group, the PCFG whales, are actually smaller than the ENP whales.
All right, question in the room.
Thanks. Great presentation, Leigh. And I only had about 15 questions. Unfortunately,
Everybody else
a
can
couple
leave.
of people have addressed some of them. I mean, you spoke a little bit about the, whether or not they might qualify as a distinct population segment. And that's obviously what is needed for the, ESA, anything ESA related. And you also sort of spoke to the, fact that maybe it's not just, I know that there was some concern about measurements taken from whaling, that the whales themselves were stretched by being dragged up onto boats or dragged ashore or that sort of thing. But you kind of spoke to that one too. So I'm going to listen. I'm going to ask this question. About the behaviors that you saw and distinct types of behaviors, were any of those associated with specific locations or not?
Okay, great questions. So the stretching thing, yes, we've heard this, and Casey's model, so those growth curves actually account for any potential stretching that has been documented. So even with accounting for that, this like 7% potential stretching when they drag the whales back to shore is in those models. So we still see that they're significantly different. So I didn't mention that. There's a lot that's in those models that I skipped way over. So your second question about behavior relative to habitat, and yes, definitely. And that is where Clara is going now. We've developed this amazing, really fine scale habitat map of our study area. And that is one major question we're trying to get at is actually we're bringing it all together. So the individual, the length, the body condition, and the habitat, and trying to see what are actually the drivers and correlates of where they do distinct behaviors. So come back to Clara's thesis defense in maybe a couple of years, a year and a half, maybe. Yeah.
All right, we're going to take a couple more questions online.
are transient orca populations increasing off the Oregon coast? And if so, any impact on the grays?
Oh, okay. Well, I don't know that that data exists to answer that first one. Are they increasing, or at least I don't have that data. So I don't know, but we do. So for the Granite project, where we're trying to understand what drives stress and stress response in the gray whales, we are including a lot of different external and internal stressors. So internal stressors like reproduction or growth, and external things like vessel strikes and noise. And one of the stressors that we are looking at is killer whale presence. So we are collaborating with a colleague that does record when killer whales are detected in our study region. And we are looking at whether those events of when the whale here causes an increase in their stress level. And I'll give you a teaser that it looks like maybe. But again, we're only maybe halfway through that analysis, so stand by.
All right, questions in the room. Okay, hang on 1 second.
Thank you. Thanks for a wonderful presentation. My question's on technology. My iPhone, if I bring up photos, I can pick a picture of my granddaughter and I can see all the images of her. Do you use that technology when you identify your whales so that it automatically identifies which whale is which?
Yeah, artificial intelligence. I wish. But people are working hard at that. Not myself, but definitely it's something that we want to go to. So for humpback whales, which are a little bit more black and white, they have been able to develop an artificial intelligent algorithm to help with that matching, just like you're talking about. For gray whales, it's harder, basically, because they are so modeled, and those models actually change over time. So it does, I mean, many of the features are static, but things can overgrow and they get barnacles and barnacles fall off. So it does take a little bit more savviness, I guess, but people are certainly working on that. We have colleagues, particularly at NOAA right now, that are trying to develop that.
All right, we're gonna take one last question from those online, and then I'll get one last question from the room, but online.
Okay, are Arctic whales larger possibly due to the colder water they live in?
Yep, that is one potential theory. Yes, there is the theory, Bergman's rule, right, Casey? Oh, he's giving me a thumbs up. Yep, Bergman's rule. That basically is this pattern. It's well documented that animals that live in colder climates are larger because they need to be larger to retain more heat. So that is one of the possible drivers of this pattern that we're seeing.
All right, last question in the room. All right, hand was first.
What do we need to do, or what can we do, or what needs to happen to get this group recognized as a separate species, well, not species, but a group that needs extra protection?
Yeah, thanks for that question. What can you do? Well, you can talk to Greg. Yeah, I think talking about, recognizing that these are a particular group, that they have distinction, that to our friends that do manage these populations. So I'm thinking about colleagues that work at ODF&W and NOAA. I think one of the major things that I want to work with my partners at those institutes on is an educational campaign. So to extend our knowledge beyond this room now, to try to encourage people when you're out there boating. I mean, we love to boat, and I love crab, Dungeness crab. Crab cakes are like my favorite thing. But to do it, when you put in your crab pot, don't let it soak too long. Get it out of the water quickly, or when it's ready, don't leave it there. When you're on your boat driving around, don't go flying over the reefs where we know they're feeding. Just go 1 kilometer off, and the chance of striking a gray whale declines dramatically. So I think, there's simple things that we can do as ocean users that will reduce, impacts on these animals. And that's one thing I definitely want to work on, is to just educate our community.
you can help by sharing this knowledge.
I think that is a lovely way to end. For folks that are online, if you have questions, you can go ahead and check out the website that's up on the screen and contact the individuals that were part of that study if you have additional questions. The same is true for the folks in the room, but you also have the opportunity to possibly come up and ask a couple of questions before Leigh runs away. So one last thank you for Leigh, everybody.
Thank you. Thank you.
And for everybody in the room, thank you so much for being a part of these events. We love sharing and love having you here. So drive safe as you leave. It is still kind of nasty out there. But thank you. And we hope to see you back here for the Film Fest in the beginning of the new year. Thanks, everyone. All right, for folks online, we're gonna wind this up. Hope to see you again in the future. Thanks.